So another important part of reaction mechanisms is knowing where electrons are moving in order to keep track of, what's happening in the reaction here. So in reaction mechanisms, we commonly do what's called arrow pushing, where we used curved arrows to show how electrons are moving as we break bonds or form new bonds. So we looked at this briefly when we talked about acid-base reactions, and we saw that there was this specific type of reaction called a proton transfer. So down here we have a list of 4 different types of electron movements or arrow pushing patterns. So we should hopefully review or remember rather that proton transfers with acid-base reactions involve 2 arrows. So that's what we see here. We see our base, our B here with a negative charge, which is the head of this arrow is grabbing this proton, right? So the tail of the arrow right here is where the electrons will start, and then the head of the arrow is where the electrons end. So that's always going to be the case with this arrow pushing type of drawing. And then we see once that grabs the proton, the electrons that were in this bond, the tail is on those electrons, and then the head goes to the A here, which is our acid, which becomes our conjugate base over here. So we can see that this new bond has formed between the base and the proton, and then the lone pair, right? The other electrons have moved to the acid. So there are 4 patterns that we're going to look at in this section here. So we have the first that's called nucleophilic attack. And now that you know what an electrophile and a nucleophile are, that should become a little more clear in this section. Our second type of arrow pushing that we'll see involves loss of a leaving group. The proton transfers, which are movement of protons, right? Movement of hydrogen atoms. And then we have our last type of arrow pushing, which will show us rearrangements. So let's start here with nucleophilic attack. So first off, we need a nucleophile, right? So that could be either a negatively charged species that has a formal negative charge or a partial negative charge because of induction. So either way, we have our nucleophilic center, or nucleophilic site. In this case, it's the bromide ion. So that is able to donate a pair of electrons. So we see our tail end of our arrow right here, where the electrons begin, and then the head of the arrow at our electrophilic site. So again, our carbocation is our electrophile. It's electron deficient. So this nucleophile, the head of the arrow, where the electrons from the nucleophile begin, the head of the arrow is pointed at the electrophilic site, and then we see our product that results over here. Right, so the tail starts where the electrons are at the negative charge, the head goes where the nucleus is, where the positive charge, and then the electrons are shared in this covalent bond at the end. So this is another example of a nucleophilic attack. In this scenario, now we have a weak nucleophile, this alcohol molecule, and it has a lone pair, right? So that's where our electrons begin, the tail end of that arrow, and then it points to this electrophilic site here. This carbonyl has a C double bond to an O, and either through induction or resonance, we see that there's a partial positive site that results at our carbon atom here, and that's where the electrons are going to be ending, right? Where that head of that arrow is, at that partial positive charge on our carbonyl, on that carbon. So that's considered our electrophile. And we also have our nucleophilic attack here, represented from the nucleophile, forming this new covalent bond with our carbon atom. So that's the site of the attack there that I just circled. So our second arrow in this drawing, we could consider this like a resonance arrow, right? This arrow that's shown down here. So that's why they're showing you this resonance that's occurring and creating this partial positive site, and then the partial negative charge, or I'm sorry, the formal charge is here on the carbon and the carbocation, and the formal negative charge on the oxygen. So this is our result over here. This is what happens when this nucleophilic attack occurs. So in this last example here, we have a pi bond, which is our site of electron density. So this pi bond is where the tail of our arrow begins, and then it goes towards this sulfur atom, and again through this electron withdrawing group, this oxygen. The sulfur has a um, is the site of the nucleophilic attack. It's considered our electrophile, and this would be our result, right? After this nucleophilic attack. So the pi bond itself is the site of high electron density and therefore the nucleophile. So then we have our second arrow pushing pattern here, where we have the loss of a leaving group. So in this case, our leaving group, right, is what has left our molecule. So that's our bromine when it's bonded, but we'll end up with a bromide ion here as far as what our leaving group is. So we have this heterolytic bond cleavage where the bromine has taken that lone pair to become negatively charged, and what results is a carbocation, right? So it's took all those electrons with it in this single arrow shown here. And again, the result is our carbocation. So we only have one arrow shown in the loss of a leaving group, but at times, depending on if there is more than one pair of electrons moving, sometimes we'll see that there are multiple arrows involved in our leaving group arrow pushing pattern. So the next pattern that we'll look at is called proton transfer. So again, chapter 3, we saw acid-base reactions take advantage of this or utilize this type of arrow pushing pattern. So we have here our acid, right? Our acid would be here above the arrow, and this could be considered our base. So again, an electron donor is our base in the Lewis acid classification. So we have our carbonyl, specifically our oxygen with the lone pairs acting as a base. And then we have the tail end here where the electrons start. They're going to grab this proton here. And then our result is the proton transfer onto our ketone, our original base here. So it's become protonated. And then we can also see deprotonation of a compound, which again involves two arrows. So I mentioned the first arrow up here. I forgot to mention our second arrow, rather, the electrons in this red line here. The tail end starts, and then those electrons are moved onto a water molecule. So this is our other product that's not shown. So then, so that's with our proton transfer in the first example. The second example down here, we see deprotonation now. So again, our water molecule is here. The lone pair now on the water molecule is where the electrons will start. They will grab this proton. So the head of the arrow is on the hydrogen here. And then within this bond, this OH bond, those electrons will move on to the oxygen. So we end up with our ketone now. So we had a deprotonation. Again, we have two arrows shown for our proton transfer. So we can look at here how proton transfer is distinct from resonance. And we have here a ketone. Generally, we don't show this carbon-hydrogen bond explicitly, but since that's where a proton will be removed, um, we show it, right? So we show this carbon atom here, and it has two other protons, but we're only interested in the one that's going to be grabbed here with this proton transfer. So we have a base, right? Our hydroxide ion here. And we're going to see that again, the arrow starts where the electron density is, right? Where the lone pair is, that the head of our arrow is pointing towards this proton, and that will be grabbed, right? And a new covalent bond will form between the OH, and then these electrons here will be moved onto our carbon atom. So the first arrow, I'm sorry, the first two arrows that we see here show that, right? They show the proton transfer. So this proton's removed, and then the lone pair is moved onto the carbon atom. So what happens is that we can also show a resonance structure after this proton transfer occurs. So this lone pair on the carbon atom would commonly move in and form this double bond here, right? So we see the tail of the arrow starting at the electrons moving in to form a double bond. That's where the head is pointed. And that causes this bonded group here to move. This electrons involved in the bond will move up to the oxygen, and a lone pair will be transferred. And then we see our double bond form here, and then the electrons have transferred from the original double bond to this oxygen. So not only do we have our proton transfer showing our arrow pushing, but then we also have this resonance structure down here with our curved arrows. So our last type of pattern that we'll talk about is rearrangements. So whenever we have a carbocation that results, um, we have a general trend of stability. So the most stable carbocation that we'll discuss today is a tertiary, and that's the highest stability. Then we have a secondary carbocation, and then a primary is the least stable. So rearrangements can occur, if possible, to create a more stable carbocation. Um, and what we see down here is this term hyperconjugation, which you don't need to know all the details about. You just need to know that in general that is a term used when we talk about stabilization of carbocations. So what we see here is a carbocation site in our molecule bonded to a methyl group, which is abbreviated ME here. And if we expand that methyl group, we'll see that this electron cloud up here, this overlap of orbitals, this slight overlap of orbitals, is actually stabilizing our carbocation. So through hyperconjugation, carbocations can be stabilized. And again, we'll see this trend of tertiary is most stable, then secondary and primary being the least stable. And again, if there is a possibility of moving either a hydrogen or a methyl group, we can create a more stable carbocation. So here's that general trend in increasing stability. So we have our methyl is the least stable here, carbocation, up to our tertiary. So each of these R groups mean that there's another alkyl group, right? So carbon and hydrogens attached. So if we can, again, have a shift or a movement of either a hydrogen in a hydride shift or a methyl group in this methide shift to create a more stable carbocation, we will. So what we see in these two examples is a secondary carbocation, right? So the carbon that has the positive charge here is bonded to two other carbons. So we would call this a secondary. carbocation. And what happens is this hydrogen in red here can actually move and it will bond to the original carbocation site. And what happens as a result is that we have now one, two, three carbons involved at this cationic site, and that would be now a tertiary carbocation due to the hydride shift. In the second example, we see a shift of this whole methyl group. Again, we have a secondary carbocation, and after the methyl shift results in a tertiary, more stable carbocation. And again, some things to note are that shifts can only occur from one carbon atom to another if it's adjacent. They can't, move more than, one carbon atom away. So that's where we'll end today with the four different arrow pushing patterns that we may see. And we'll continue next time with the next section.